So we mentioned in the previous video that natural attenuation or intrinsic bioremediation is there, then enhanced bioremediation is there. Don't forget these two terms. Now moving on to the next session. Many approaches are being used effectively to catalyze or enhance bioremediation. Sometimes the simple addition of an otherwise limiting nutrient is appropriate, while at other times microbial inocula are needed. In other situations, it may be important to add readily available substrates to foster co-metabolism of a contaminant or to drive an environment towards anaerobiosis to stimulate anaerobic degradation. Sometimes it may be necessary to add surfactants or other chemicals to increase the bioavailability of the contaminant. So sometimes it may be necessary to add surfactants or other chemicals to increase the bioavailability of a contaminant. To sum up, we may say that fundamentally there are two approaches to enhance bioremediation, like biostimulation, which depends primarily on the modification of the environment, and then bioaugmentation, which uses the addition of microbial cultures to increase biodegradation. So biostimulation, where we use the existing environmental conditions, and in the next stage or the next method of bioaugmentation, which uses the addition of microbial cultures to increase biodegradation. Frequently, these two approaches are combined and microorganisms are introduced in conjunction with such environmental modifications as nutrient supplementation. Bioremediation therefore overlaps several older biotechnologies. Municipal and industrial wastewater treatment is a well-established industry, and while it can be distinguished from bioremediation in that the pollutants are under physical control during treatment, many of the fundamental biological processes are the same. Similarly, composing is a well-established phenomenon currently gaining popularity in the municipal solid waste treatment industry, and the biofiltration of waste gases is becoming a useful technology. Development of these technologies for cases in which the contaminant is already under physical control will undoubtedly aid the development of bioremediation as an accepted tool for dealing with similar wastes when they have escaped our control. For bioremediation of polluted waters and soils, the process can be carried out in place, like in situ bioremediation, or after the contaminated material has been removed or moved to some sort of contained reactor. Ex situ bioremediation, in situ and ex situ. The former is generally rather than less expensive, but the latter, that means ex situ, we are expected to move that particular setup into a contained reactor. That is meant by the ex situ bioremediation. I am not sure about the pronunciation, but pardon me. The former is generally rather than inexpensive, but the latter may be much faster than the additional cost of manipulating the contaminated material is overshadowed by the time saved. Then ecology and bioremediation. The biosphere plays a major role in the great element cycles of our planet, and bioremediation must be placed in this context as if we are to appreciate its broadest ramifications. One of the underlying fundamental truths of biological diversity is that if there is free energy available in the metabolism of a substrate, there is probably a guild of organisms that has evolved to make use of it. This is particularly important for the biodegradation of organic molecules. For example, crude oil seeps to land and water have occurred for millennia, and as a consequence, aerobic oil degrading microorganisms are developed. If biology does not yet take advantage of a source free energy, then we can expect that there will be strong selection pressure in favor of any organism that develops an ability to exploit it. This has been seen with byproducts of nylon manufacture Pseudomonas aeruginosa was gained the ability to degrade the novel compound 6-aminohexaanone 8 linear dimer, a byproduct of nylon 6 manufacture, as the sole source of carbon and nitrogen. The successful bioremediation of xenobiotic compounds such as pesticides and herbicides may sometimes present a similar acquisition of genetic traits. However, not all organic molecules provide a source of free energy. Some, such as small halogenated solvents, provide no significant source of nutrient or energy, and their aerobic destruction can only occur co-metabolically with the degradation of a more nutritious substrate or anaerobically as a terminal electron acceptor. This white rot fungi offer another variation of this theme. These organisms seem to be unique in their ability to degrade lignin, the structural polymer of higher plants. They may not gain any direct energetic benefit from lignin degradation, but it clearly allows them access to cellulose, which is a substrate for their growth. Lignin degradation is catalyzed by a group of extracellular peroxidases that generate non-specific oxidants, and there have been several proposals to use these systems for destroying contaminating organic compounds. With successful bioremediation, organic compounds are eventually converted to carbon dioxide, water, and biomass. Similarly, nitrogenous molecules such as excess ammonia or nitrate in molecules such as excess ammonia or nitrate with groundwater can be converted to gaseous nitrogen. Other non-organic contaminants provide a different challenge for bioremediation. A few such as mercury and selenium are volatilized by some biological process, but it is not clear that this is always beneficial. In some cases, just chromium and arsenic, there is a dramatic difference in the environmental toxicity depending on the redox state of the contaminant. In other cases, bioremediation has sometimes focused on this detoxification by bacterial processes. A more satisfying approach would be to use a biological process to accumulate and concentrate the contaminant so that it can be removed for safe disposal. Fungi, algae, and higher plants have all been used in these efforts. Technology of bioremediation, what is happening in that? Successful bioremediation hinges on the effective application of biology discussed above. Sometimes the contaminant is on the surface, so access to it is reasonably simple. In these cases, the required technology may be as simple as broadcast spreaders or sprayers to apply fertilizers or tilling the soil to allow aeration. Of course, it is not necessarily as simple as it sounds, since contaminated sites are very often different from agricultural fields, and the technology has to be significantly stronger to plow the soil. Frequently, the contaminant is below the surface. The applying even small or simple bioremediation strategies provide quite a challenge. Foci of R&D for bioremediation. 3 different foci of R&D for bioremediation research have emerged worldwide. While Europeans are expanding their traditional waste and water treatment systems to cope with specific chemical pollutants, Americans focus on site-specific cleanup of soil and water contaminated with petroleum and xenobiotics, and the Japanese take aim at global environmental problems. Each of these 3 technologies of bioremediation in environmental cleanup will be considered in somewhat detail below. European focus: upgrading traditional waste and water treatment systems. Several European countries, particularly Germany, Netherlands, Belgium, Australia, and Italy, have programs to foster R&D in environmental biotechnology. German government agencies promote technology transfer and cooperative research between government-sponsored research institutes and private industry. In the Netherlands, the most developed country in environmental biotechnology, the government supports an innovative program in bioremediation R&D and subsidizes industries participating in the program. For example, long back in 1978, the Dutch Nuisance and Air Pollution Act took advantage of the development of biofilters, a modification of traditional trickling filters, permitting installation of these biofilters to meet air quality standards. Following are examples of the developments made in the waste and water systems in some European countries. Biogas energy from solid waste and refuse. It could be recognized by some newly developed European waste treatment systems that some microbial degradation or transformation occur only in the absence of oxygen, and such a process could be important in environmental biotechnology. Under anaerobic conditions, microorganisms growing on wastes can sometimes produce valuable fuels. The dry anaerobic composing Dranko process converts the organic fraction of biodegradable organic solid waste and refuse into energy in the form of biogas, methane and carbon dioxide, and a humus-like material. The biogas is produced by a consortium of anaerobic bacteria that includes methanogens, methane-producing archaebacteria. Full-scale installations using Dranko process are in operation at Belgium and Austria. The systems treat separated plant residue and paper from municipal waste. In Austria, both aerobic and anaerobic microbes are used. The first phase of treatment, anaerobic digestion, take place in a huge tank lasting for about 3 weeks. The digested material then moves for aerobic maturation, which takes about 2 weeks. The process is very important. The facility built by Organic Waste Council of Belgium can treat 20,000 tons of bio-waste every year. Such contaminated anaerobic and aerobic treatment systems replace traditional landfills for disposal of solid wastes. These new systems produce biogas that is about 55% methane, which is burnt to produce electricity. Removal of inorganic compounds. Some waste filter systems, originally designed only to remove organic compounds from water aerobically to reduce BOD, have been modified to induce anaerobic zones to remove inorganic compounds as well. One such system, a pilot-scale fluidized bed reactor that removes nitrate from water, has been tested at the municipal water treatment facility in Blanchard, Belgium. The removal of the nitrate from wastewater helps prevent eutrophication of the waterways receiving the treated water. At Blanchard, nitrate concentration was reduced from 75.0 mg per 1 to 0.1 mg per 1 when water was passed through bioreactor. The reactor contains the methyl trophic bacteria such as methylotrophis that carry out denitrification. Methanol is first added to bioreactor to support the growth of methylotrophis and later removed by trickling filters and granular activated carbon filters before the water is discharged for use. The bacteria convert nitrate to nitrite and then to molecular nitrogen, which is released into the atmosphere.